Hello, my name is Susumu Goto from Kyoto University, and thank you. I'd like to thank the organizers for inviting me to present our work. And we are developing a database named KEGG, which includes not only the metabolic networks, but other protein-protein interaction networks. But I would just to talk about metabolic networks. So I'm going to focus on the metabolic networks. But in fact, we do not use any state-of-the-art visualization techniques. But I would like to talk about how we are creating the database. So today's my talk, I would like to organize by asking 3 questions to myself. And the one is how do we visualize the metabolic networks, which is a sort of the methods we use. And the second one is why do we visualize them in that way? This is a sort of the objectives. And the last one is what can we see by visualizing the metabolic network in that way. This is a sort of results from this method. So I'd like to first talk about why do we visualize. And this is a traditional metabolic networks. This is actually the same one the previous speaker showed in his slides. And this is developed by, I think, the Boehringer Mannheim and currently a Roche pharmaceutical company. And this is very famous wall chart for the biochemist, indicating the whole metabolic networks in our knowledge. So what this shows is the metabolic network is a flow of the chemical compounds, which is a biosynthetic pathways or biodegradation pathways. And this also shows the consecutive enzymatic reactions. So you can see which enzymatic reactions work together to produce some biochemical compounds. And another important issue is the layout of these networks. Actually, many biochemists or molecular biologists or biologists get used to this kind of layout, such as in the center there is a glycolysis, and then the TCA cycle are represented in the cycle or something like that. So to represent this kind of metabolic network in the computer, we had basically the 2 concepts. One is a hierarchical visualization. We would like to represent the whole network, like the previous slides, to the reaction itself in the computer for some analysis. And the second one is 2 aspects of the enzymatic reactions. So we can see each enzyme reactions as just a chemical reactions, but we can also see the enzymatic reaction as gene products because each enzyme are coded by the gene. So the first one is a hierarchical visualization, and we organize our database into roughly the 6 levels of the abstraction. The top level is overview, like the previous slides, and the bottom is molecular reactions or molecules or chemical compounds. So I will show you each of them one by one. So the first one is the overview in this KEGG pathway diagrams is like this. And this is a whole view of the network, of course. And this shows a network of chemical compounds and the reaction sets. So each small dot represents the chemical compounds, and the line between the small circle dot represents the reaction set. So we put several reactions which catalyzes the enzymatic reactions between the two compounds as a reaction set. And we color this network based on the functional categories, functional classification. So this network is drawn basically manually by KEGG Sketch, which is an in-house Java application which we use for drawing this kind of diagram. So this is just another drawing software, but which can also associate the objects in this diagram to the objects in the database we are developing, such as a chemical compound database or reaction database or some other genes database. And the second level describes a pathway diagram like this. And if you use the KEGG database, maybe this is the most popular or familiar with you. And this is also the network of the chemical compounds and reactions. But in this case, each box represents one reaction. And this diagram shows the functional subsets of the whole network. And this subset is defined based on the biochemical knowledge. And this diagram is also drawn manually by the KEGG Sketch. And the third level we define is a module, which is a subset of the pathway diagram, and which indicates more functionally tightly related set of the reactions or enzymes. So this is a functional subset of the diagram and networks of the chemical compounds and reactions again. And this is defined based on the biochemical knowledge and also the genomic context, such as operon structures in the bacteria or the phylogenetic conservation, like this part is only for the eukaryote, this part is only for the archaea or something like that. And this is also, this is manually defined, but automatically drawn by coloring this diagram like this, or just align this enzyme or post-log box like this from the start point of the module to the end point of the module. The fourth level is a reaction, and this is just a chemical reaction, and represents the conversion of chemical compounds. And we define the main metabolites and cofactors like like this one and this one for the main metabolites, and this one the cofactors, basically based on the KEGG pathway diagram. So these are manually defined and automatically drawn using the chemical structures we stored in the database. And the fifth level is the molecule. I show this here the chemical compound, but in this case, the molecule can be an enzyme or genes, which represent the reactions too. So we use 2D chemical structures like this, including some chirality or stereochemistry. And we draw these 2D structure manually by KEGG Draw, which is another in-house Java application to draw the 2D structure of this chemical compound and also the carbohydrate structure. And the last one is reaction patterns. And this is a bit different from these molecules and the pathways, but this is the subset, sort of the kind of subset of the reactions. And this is also manually defined and using the reactions, mainly from the main metabolites. And those main metabolites and reactant pairs are automatically aligned and manually modified to draw the reaction patterns like this, which indicates reaction centers, in this case the red part. So these two carbons are reaction centers, and the blue part indicating a changing part of these reactions. So those are the hierarchical visualization. I mean, hierarchical classification of the data in the KEGG databases. And another aspect is, another concept is the two aspects of the enzymatic reactions. So each pathways include rectangles like this, indicating the chemical reactions and also gene products. So each rectangles in this pathway diagram represents the EC number like this, which are defined by IUBMB enzyme committee, and also represents a chemical reaction like this. So we created the reaction database, and we assigned each reaction ID to this box. And in terms of the gene product, we can also assign the gene ID. In this case, this is a human gene entry to this box, and also the ortholog IDs, which are the definition of the collection of the genes which have the same function into this box. So we can use this ortholog ID to reconstruct the species-specific pathways. And by using this gene product, we can see this pathway diagram as a network, not only for the chemical network of the chemical reaction, but also the network of enzyme genes. So the next, I'd like to talk about the why do we visualize them in this way. As you know, we have a lot of genome information, transcriptomic information, proteome information, and metagenomic information, so-called omics data for the genetic information. And we also having a chemical information in terms of omics data, such as metabolomics information, the lipidomics, or glycomics information. And those rapidly increased genomic and metabolic data, we have to interpret somehow them. So to do that, we need some current knowledge of the biologist or biochemist or molecular biologist, and which we call the systems information. And these information can be pathways or function or disease, something like that. So by accumulating the systems information, we would like to easily reconstruct the metabolic or other networks from the genomic information. We also would like to easily interpret omics data. And we also would like to predict some new metabolic networks which are not stored in the database yet, but there should be. So to do that, the integration of these metabolic and genomic information is necessary, and also the systems information should be integrated to this information. So the KEGG database, so we are developing a KEGG database. Actually, we are currently creating 19 databases and in the KEGG database suite. And these databases are categorized into the 3 parts. One for the knowledge on the systems, like a KEGG pathway database, KEGG bright database for the functional hierarchical classification of the gene functions of chemical compounds, and knowledge on the genome, and the knowledge on the chemicals to link the biological knowledge to omics data, such as the genomic and the metabolomics information. So by this database, first we can automatically reconstruct the metabolic network by using the gene product aspects of this pathway diagram. Because once we get the genomic information, we can assign the gene function of the genome by using such as homology search or the motif search. Then we can assign the ortholog ID to each gene in the genome, and then mapping these annotation, functional annotation, to the networks by coloring like this. So in this case, I show the human glycolysis pathway, which in which the green color indicates the enzymes the human actually has, and the white box indicates the enzymes the human does not have. So we can see the green part is a human pathways. And in terms of the interpretation of omics data, we can also use this relationship, because we can map the transcriptomic information and the proteomics information to the KEGG genes database information, and the metabolomic information to the KEGG ligand chemical part of the KEGG database, and then map those information to the KEGG pathway diagrams like this. In fact, you can do this for the whole overview of the metabolic networks like this. And I'm sorry, we still have a problematic green and red color link for the microarray data, but what I'd like to show is you can map both the transcriptomic and the metabolomic data into the pathway diagram, so that you can see the relationship between not only for the transcriptomic data itself, but the relationship between the transcriptomic and the metabolomic data. And I'm not going into detail the pathway prediction, but once we define the reaction patterns I show at the very bottom level of this representation, visualization, you can predict some new pathways from a new compound which are not stored in the database yet. So in this case, this is a query compounds. Actually, this is environmental chemicals, or you can use the plant secondary metabolites for the biosynthetic pathway prediction. And then this system map this new compound to the known reaction patterns like this, and then convert the this query compound into the new compound based on these reaction patterns. So by repeating this step, you can somehow predict the new pathways. So the last part I would like to show is what can we see by visualizing them, visualizing the metabolic network in this way. As I told you, we can easily reconstruct the species-specific metabolic networks, and we can also map the omics data to the pathway database or bright database for the functional interpretation of those omics data. But I'd like to add one more thing, which is multiple species mapping at once. So you can use not only one species, but the several species to visualize pathways like this for looking at the relationship between the symbiont and host, and pathogen and host, or the human and its gut metagenome. And in this specific example shows the symbiont of the P. aphid, which is an insect, and Buffonella bacteria, which is an aphid symbiont bacteria. And it is well known that these two species help each other to produce several amino acids, in this case, valine, leucine, isoleucine. And as you can see, the three amino acids is here. And as you can see, to produce these amino acids, both the green part and the pink part are necessary. And the green part shows the enzymes that the only P. aphid has, and the pink part is only the enzymes only have only the Buffonella has. So you can see these two species have to help each other to produce those amino acids. And the very last part here is enzymes which produce proteins from the amino acids. So both species have those enzymes. And you can see again the whole view of this multiple species pathways. And in this case, the green part is for the P. aphid, and the red part is for the Buffonella, and the blue part is for the common enzymes pathways. And here is the amino acid metabolism part, and you can see both the red part and the green part are connected to each other. And this is very well known, but our collaborator noticed that here is also the connection there is a connection between the red part and the green part. And this is actually the nucleotide metabolism pathway. And they are currently investigating what is going on here. So I'd like to summarize my talk here. So the first question, how do we visualize the metabolic networks? We use a hierarchical representation from whole networks to the molecules. And we use the two aspects, and the chemical reactions and gene products for each enzymatic reactions. And why do we visualize them? We would like to connect the chemical metabolic reactions to the genomic context. And to do that, we can also use them as a omics data analysis. And another important issue is a layout of the objects and the connections in the pathway diagram. So we basically use a manual drawing for that pathway diagram. And what we can see by visualizing them in that way is multiple species at once, of course. And we also, the KEGG database is still expanding. So we define the other networks, some metabolisms like protein-protein interactions or gene disease networks or developmental history of the drugs. But I would like to, we keep our database simple as much as possible, but still informative. So I'd like to thank all the KEGG project member and the system engineer, and also the funding agency. I'm sorry this is in Japanese, but the Japanese funding agency. Thank you for your attention.